Okay, guys, quickly, I want to run a series of classified paper for IGCSE biology. I think I've done for chapter 2, I've done for chapter 1, so let's quickly have a look on chapter 2, which is organization of living organisms. Here, the first question I have here is that said, a biologist made a slide of some epidermal cells from a scale leaf of an onion bulb. Now, figure 4.1 is a drawing that the biologist made of one of the cell. Now, they said the table 4.1 shows the function of the structures within a plant cell. So complete the table by naming the part of the cell that carries out the function and using letters from figure 4.1 to identify the part of the cell named. Quickly, before doing that, let me identify this part. Obviously, C is the nucleus. B here should be the cytoplasm. A, since it's an onion bulb, obviously you know that A is your cell wall. And sorry, D is the cytoplasm. Why this B here is your vacuole. So if that's the case, they say resist the toggle pressure of the cell. That should be the cell wall. And the letter that represent that is A. Now they say control the activities of the cell. That's the nucleus, and that's C. Site for chemical reaction to take place. That's the cytoplasm, and that's letter D. Okay. Now, figure 3.1 shows image of red blood cells from a human A and a bird B. So this is red blood cells from human A and bird B, and in bird B we are told that there is a nucleus. Now, state the function of red blood cells. Simple, they help to transport. Say one mark question, transport oxygen around the body. Now, there is a nucleus present in each of the red blood cells for birds, shown in figure 3.1. State the function of the nucleus. Now, nucleus contain genetic information, and controls cell activities. Now, human red blood cells do not contain nucleus. Yes, state the advantage of this. So, you can just write to carry more hemoglobin to transport oxygen. That's all. They say leaves are made from tissue. Define the tissue. Tissues are a group of cells that carry out similar function. That's it. Now they said figure 4.1 is a photograph of a transverse section of a leaf showing the upper and lower surface. Now, this is the upper surface. There's a leaf hair on the upper surface. So beneath the upper surface, your B is the palisade. Mesophyll cells. Then if this is the stomata here, root hair cell, then A here should be your spongy mesophyll cells. So name the tissue A spongy mesophyll tissues. The process by which gases travel through the stomata, gases move by diffusion from an area of high to an area of low concentration. Describe how the root hair cell differ from cell B. Cell B is the palisade cell. How does it differ? Okay. First, you know that root hair cells do not have nucleus. Root hair cells do not have nucleus, but cell B do. That is palisade cell. Another thing there is root hair cell is elongated to give the root a large surface area in shape, while the upper one there, cell B, is like columnar in shape. That will give you a full mark. Now, figure 1.1 shows a common emerald dove. This is the scientific name. Two distinguishing features of birds are beak and wing. State one other features shown only by bird that is visible, what you can see. It has feathers. That's all. Now, I have a question here too. It said the boxes on the left contain the names of cells and tissues specialized for carrying out a particular function. And the boxes on the right is the description of specialized function. Draw a straight line. Draw a straight line to link each specialized cells or tissue with its correct function. An example has been done for you. So okay, so the first one, ciliated cells, they help in movement of mucus around the trachea. Root hair cells help in the absorption of water. Red blood cells help to transport oxygen around the body. Xylem vessels help to transport water. Remember, root hair cells do not transport, they absorb water and mineral ions. You should take note. Egg cells is for reproduction, while white blood cells is for defense, so it undergo the process of phagocytosis. Now, figure 4.1 is a diagram of a plant cell, and they say identify the part of the plant cell in figure 4.1 by labeling them in the spaces provided using words from the list. Okay, so using these words, you find out that this should be a cell wall. Then this will be the cytoplasm. This will be the chloroplast. And this will be cell membrane. For sweet mark. Now, they say the boxes on the left show part of a plant cell, while the boxes on the right shows the function of this part. Draw a link to their function. Now, cell wall is for made from cellulose and strengthen the plant. Chloroplast is site for photosynthesis. Nucleus contain genetic materials. Vacuole filled with cell sap to support the plant cell. Now, the boxes on the left describe processes carried out by plant cells, while the boxes on the right contain the name of the cell that carry out these processes. Draw one straight line from each box on the left to a box on the right to link the processes to the cell type. Draw 6 lines. Wow. An example has been done for you. 6 lines. Okay. Absorption of mineral from the soil, root hair cell, antibody formation, white blood cells, blood clotting, platelets, production of glucose, palisade mesophyll cells, because they also help in photosynthesis and produces glucose, phagocytosis, white blood cells, transport oxygen, red blood cells. So how many lines did I draw? 1, 2, 3, 4, 5, 6. So that's the answer. Now, yeast cell have many structures in common with a plant cell. Okay, figure 4.1 is a drawing of a yeast cell. So if this is it, then you know your B. I always advise you label them first. It's your nucleus. Your C here is the mitochondrion. D is the vacuole. Cell wall. Okay, my A here is my area, rough endoplasmic reticulum. Then state the name of the cell structure label A and D. A, area, D, vacuole. State the function of the cell structure label B and C. B contain genetic information, C for respiration to release energy. Contain genetic information so it can control cell activity, and C for respiration to release energy. Now, state the name of one structure that is found in a plant cell but absent in a yeast cell. This doesn't have plant cell but absent in a leaf cell, so a chloroplast is absent there. Now, arrange the structure in the list in increasing order of organization, starting with the smallest structure. The first one has been done for you, obviously, from cell to tissue, from tissue to organ, from organ to organ system, from organ system to organism. Now they say the boxes on the left show different parts of a flowering plant. Now the boxes on the right are terms that describe the different level of organization. Draw one straight line from each part of the flowering plant to its level of organization. The flower should be this. The flowering plant is an organism. Palisade mesophyll cells. Root hair cells, palisade mesophyll tissues. Because they are a group of, if you look at it, they are together, that makes it a mesophyll tissue. Okay, now they say Baker's yeast, this is the name, is a cell is a single cell organism that is classified in the kingdom fungi. Now figure 1.1 is a drawing of a section through a yeast cell. So state if you can just label this first. This is the cell wall, this is the cell membrane, vacuole, cytoplasm, mitochondria, area, rough endoplasmic reticulum, nucleus, ribosomes. Okay, if this is it, this is state one other kingdom that contain organisms that have structure A, cell wall, that's plant. Now they said table 1.1 shows some cell functions. Complete table 1.1 by naming the cell structure responsible for each cell function and give a later storage of gene. It's the nucleus. Aerobic respiration, mitochondrion. Assemble amino acid where protein is made is the rough endoplasmic reticulum, or you can put the ribosomes. So the letter that represents nucleus here is G. So nucleus is G, mitochondrium is E, rough endoplasmic reticulum is F, and ribosomes is H. So you can, any of those two we go. So I can put my F or my H, whichever. Now a student made a drawing of one oh, okay, yeah, I just underlined the word, pronouncing it sounds so funny. Bacterium. Now figure 1.2 shows the student drawing. Okay, now the actual length of the bacterium is these micrometers, and they need to convert to millimeters. You know, from millimeters to micrometers, you multiply by 1000. So obviously from micrometers to millimeters, now you're going to divide by 1000. So your answer will be 2 divided by 1000. State the other information that the student need in order to complete the magnification of the drawing. Now you have actual, remember magnification, M is equal to image size over actual size. So he has actual now, what he needs is the image size. Describe the similarities and differences between the structure of the yeast cell and the structure of the bacterial cell. So this is the yeast cell and this is the bacterium cell. So what you should do also, label it. This will be cell wall. This is ribosomes. This here is cytoplasm. This is DNA or RNA. So yeah, if you want to look at, I can decide to write similarities and my differences. Similarities is that both have cytoplasm, both have cell membrane, Both have ribosomes. Both have DNA. Mhm. Now, differences is find out that yeast have nucleus. Yeast have mitochondrium. Yeast have rough endoplasmic reticulum. Yeast have a vacuole also. Yeast have a vacuole. This will give you a 6 mark. Now, figure 1.1 shows some cells from the lining of the trachea. Okay, now describe the function of the nucleus and cell membrane. This is quite simple. Nucleus contain genetic information, and you see this is a 4-mark question, so need more detail. Contain genetic information. Contain genetic information to control cell activities. Cell membrane control the movement of substances in and out of the cell. In and out of cells, which makes it it is semi or partially permeable. Now, define a tissue. I think we've done this earlier, where we say tissue are a group of cells that have similar functions. Now, describe the role of mucus and cilia in the trachea. So we're looking at two things: role of mucus and cilia. Mucus, this is not chapter 2 though, but mucus help to trap dust particles in air and prevent it from reaching the gas exchange surface. Gas exchange surface. So cilia help to move the mucus in the trachea to the mouth, where you either spit it out or you swallow into your stomach. That's it. Now, figure 1.1 is a photomicrograph of a leaf of a tea plant. Kind of question come up. No, this is the palisade mesophyll tissue, because you're looking at a group of all of them. Mesophyll tissue. Why this is S space, and this is your stomata. This is the lower epidermis. Now, name, I just named them. So just write everything there. Okay, this is the, you can call it the waxy cuticle on the surface. So just label that. You're good. Now, the figure 1.2 shows a cell from region B of, okay, that's the palisade cell. F here is your nucleus. G is the cell membrane. This is your cytoplasm. J is the cell wall. K is the vacuole. L here is the chloroplast. Use the later from that table. Control movement of substance in and out of the cell is the nucleus. Exact pressure, vacuole. Produce sugar, chloroplast. Withstand the internal pressure of the cell, that's cell wall. Control the activities of the cell. Control movement of substances in and out. Cell membrane. Control all activities in the cell, that's the nucleus. That's all. So if you come here, I've labeled them so you can just pick out the letters. I think we've done similar stuff. Okay, here they said figure 2.1 shows a photomicrograph of bronchus surrounded by alveoli and other tissues in the lungs. Okay, what kind of question is this? Magnification. Okay, when you're given a question like this, the first thing you need to do is use your ruler. The instruction will be given to you. This type of question comes up more in paper 6. They said the diameter of the bronchus is 2.1. In figure 2.1 is shown by letter A to B. Measure the length A to B on figure 2.1. Include your unit. So our advice to most students is try as much as you can to make sure your measurement is in millimeters. So you get a ruler, length of AB. Let's say you use your ruler to measure this length and you have 5 centimeters. You can change it to millimeters when you multiply by 10. That will give you 50 millimeters. So if you have 50 millimeters, in this case, you come put your 50 millimeters. That's length of line AB. Now the actual diameter of the bronchus is this. Now they said the magnification calculated. Length of line AB in your diagram that you measured is 50. Divide by actual diameter, which is 1.5. Remember, this is millimeters, this is millimeters. So this cancel out this. So anything you get, you put times in front. Once you divide, you get your answer. That's the magnification. That's it. I think this should be similar. Figure 2.2 shows a different kind of flower. Okay. Measure the length of the label anta on the photograph. Okay, that they want you to measure this length. You can measure from here to here. Let's say you have 2 centimeters. You convert, you times it by 10 to give you 20 millimeters. So you come put 20 millimeters. They now said use your answer to figure 2pr and the equation to calculate the actual length of the anta. So if you look at this figure, the magnification is times 2. So magnification here is given. We have our length of anta. We need to calculate actual length. So magnification is 2 is equal to length of our anta, which is 20, divided by actual length. So this will now, if you cross multiply, you now have actual length times 2 is equal to 20. So your actual length is equal to 20 divided by 2. So my answer here will just be 10. Okay, so figure 2.1 is a drawing made from an electron micrograph of a lymphocyte that produces antibodies. A lymphocyte that produces antibodies. Okay, so if you want to label this, which is the first thing I advise you to do, is and this is your cell membrane. This is the nucleus. This is a single dot. It's touching a single dot there. That makes it the ribosomes. This is the cytoplasm. This is the mitochondria. This is small temporal vacuole or vesicles, while this is rough endoplasmic reticulum, because it's looking at all of them. Now, the set of the 2.1 contains statement about the structure visible in figure 2.1 completed. One has been done for you. Store of genetic information. Obviously, you know genetic information as DNA stored in the nucleus. Provide energy, that's for making antibodies. So provide energy through respiration is the mitochondria. Site for production of antibodies. Antibodies is an example of protein, so it's similar to site for production of protein. So that can be a rough endoplasmic reticulum, or you can call it the or also the ribosomes carry out that main function. Transport of antibody molecules for release into the blood. Transport. If you look at this, you find out that the vesicles are the ones moving substances from inside and coming out to the cell membrane. So I'll give that to the vesicles. So to get your remaining 4 mark, you just label the path. Nucleus, you come, is B. Your mitochondrium is E. Your antibody production, rough ER, is G and C. So G/C, and vesicles are F. That's it. Now, so figure 5.1 shows a diagram of a cross section of a leaf, and okay, so this is the palisade mesophyte tissue, because you're looking at all this group of cells, and this is your guard cells. So palisade mesophyte tissues, and these are guard cells. Controls the opening and closing of stomata. Now, the cell in tissue A are adapted for photosynthesis. State where photosynthesis occur. Photosynthesis occur in the chloroplast. Now, they said draw circles around the structure that are found in both plant and animal cell. So both plant and animal cell have cell membrane, both have cytoplasm, both have nucleus. So I think these are the main type of questions that comes out in paper chapter 2. The most of the question, there are some, it's a mixture of both core and extended. That's what I just went through. So you can watch, put your comments in the comment section, and if you have any chapter that you have issues with and you want me to treat questions on that particular chapter, you can just put it in the comment section and I will make sure it is done.